Hello students, today's topic is aflatoxins. So, aflatoxins are one of the major group of mycotoxins, and the aflatoxins are poisonous carcinogens and mutagens that are produced by certain moles which grow in soil, decaying vegetation, hay, grains, etc. And they are mainly produced by Aspergillus flavus and Aspergillus parasiticus. They are regularly found in improperly stored staple commodities like cassava, chili, pepper, peanut, rice, sesame seeds, sorghum, etc. So, aflatoxin is an example of mycotoxin. We know that mycotoxins are the toxins which are produced by the pathogenic fungal species. And one major example of the mycotoxin is the aflatoxin, which is produced by the Aspergillus species. And aflatoxins are poisonous carcinogenic compounds. They are poisonous carcinogens and also they are mutagens which are produced by Aspergillus species, and mainly they are found in soil, in the decaying vegetation, hay, grains, etc. And examples of the Aspergillus species for producing these aflatoxins are the Aspergillus flavus and the Aspergillus parasiticus, and they are regularly found in improperly stored food items like the cassava, chili, pepper, peanut, rice, sesame seeds, sorghum, etc. And coming to the types of aflatoxin, first one is the aflatoxin B1 and B2, which are produced by the Aspergillus flavus and the Aspergillus parasiticus. Next one, aflatoxin G1 and G2, which are produced by some species of Aspergillus flavus and the Aspergillus parasiticus. That is a group 2, Aspergillus flavus and Aspergillus parasiticus. And next is the aflatoxin M1, which are the metabolite of the aflatoxin B1 in humans and animals, and aflatoxin M2, which are the metabolite of aflatoxin B2 in milk of cattle fed on contaminated food, aflatoxicol, which are the metabolites produced by breaking down the lactone ring, aflatoxin Q1, major metabolite of aflatoxin B1 in vitro liver preparation of other higher vertebrates. So, coming to the different types of aflatoxins, there are mainly 6 types of aflatoxin. First one is the aflatoxin B1 and B2, which are produced by Aspergillus flavus and Aspergillus parasiticus. Next one is the aflatoxin G1 and G2, which are produced by some group 2 members of the Aspergillus flavus and Aspergillus parasiticus. So easy to remember. First one is a B1 and B2, which are produced by Aspergillus flavus and Aspergillus parasiticus. Next one is the G1 and G2. They are also produced by some group 2 members of Aspergillus flavus and Aspergillus parasiticus. Next one is the M1, aflatoxin M1, which are the metabolites of aflatoxin B1 in humans and animals. So, aflatoxin M1 are the metabolites of the aflatoxin B1 present in humans and animals. Next, after M1, aflatoxin M2. They are also metabolites of the aflatoxin B2 in milk of cattle fed on contaminated food. So, first B1 and B2, next G1 and G2, M1 and M2 are the metabolites of aflatoxin B1 and aflatoxin B2 respectively. After that, the next type is the aflatoxico. It is also a metabolite which is produced by a breaking down of the lactone ring of the aflatoxin. And the next one is the aflatoxin Q1, which is a major metabolite of the aflatoxin B1 in vitro liver preparation of higher vertebrates. So, aflatoxin is a major metabolite of the aflatoxin B1 in vitro liver preparation of the higher vertebrates. Coming to the occurrence of this aflatoxin in food item, you know that Aspergillus is one of the important foodborne pathogen, and also the factor that make them more severe is their ability to produce this mycotoxin, that is their ability to produce aflatoxin. And coming to their occurrence in food, they are mainly found in various cereals, oil seeds, spices, nuts, etc. And also they contaminate grains and cereals at various steps during the harvesting or storage. And aflatoxin contamination of wheat or barley commonly occur due to inappropriate storage. And in milk, aflatoxin is generally present at 1 to 6% of their total content, and the aflatoxins infect humans following consumption of the aflatoxin contaminated foods such as egg, meat products, milk products, etc. So next topic is the occurrence or the chance of presence of aflatoxins in food. In which type of food these aflatoxins are commonly present, or what are the major kinds of food which these Aspergillus species will spoil. They are mainly found in various cereals, oilseeds, spices, nuts, etc. And also they will contaminate the grains and cereals which are during the various steps during their harvesting or storage. There is a chance of contamination of grains and cereals at the various steps of the harvesting and also at the storage step. And the aflatoxin contamination of the wheat or barley commonly occur due to the inappropriate storage. So, the occurrence of aflatoxin producing fungus or the production of aflatoxins in wheat or barley products is mainly due to the, or is mainly because of the inappropriate storage process. And in milk, aflatoxin is generally present in 1 to 6% of their total content, and aflatoxins infect the humans following consumption of the aflatoxin contaminated foods such as egg, meat products, milk and milk products. So this aflatoxin can enter into humans and lead to foodborne illness through the food like contaminated food like egg, meat products, milk products, etc. Coming to the detection techniques of aflatoxin, aflatoxins are usually detected and identified according to their absorption and emission spectra, with the peak absorbance occurring at 360 nanometer, and the B toxins exhibit blue fluorescence at 425 nanometer, while the G toxins show green fluorescence at 540 nanometer under UV radiation. And this fluorescence phenomenon is widely accepted for aflatoxins, and thin layer chromatography is among one of the oldest techniques which is used for the aflatoxin detection, while high performance liquid chromatography or HPLC, liquid chromatography, mass spectroscopy or LCMS, and enzyme linked immunosorbent assay or ELISA are the methods which are commonly used for the detection of aflatoxin. ELISA can also be used to detect aflatoxin or used to identify which type of aflatoxin is present. And coming to the detection techniques, the aflatoxins are usually detected and also identified according to the absorption and emission spectra, and also with the peak absorbance occurring at the 360 nanometer. And aflatoxin B or the B toxins exhibit blue fluorescence at 425 nanometer, whereas G toxins show green fluorescence at 540 nanometer under UV radiation, and this fluorescence phenomenon is widely accepted for the detection of the aflatoxins. And TLC, HPLC, LCMS, ELISA, etc. are the techniques which are used for the detection of the presence of aflatoxin in a sample. And the ELISA can also used to identify the type of aflatoxin. Then coming to the management and control strategies, as we all know, the aflatoxin producing fungus species, that is a contamination of the food item with Aspergillus species, is very harmful and it will lead to many foodborne illness or foodborne diseases. So it is essential to control the occurrence of Aspergillus flavus, or the spoilage of the food item by Aspergillus flavus, and also the production of aflatoxin or mycotoxin in the food items. Then coming to the management and control strategies, the use of biocontrol agents like Bacillus subtilis, Lactobacillus species, Pseudomonas, Ralstonia, Burkholderia, etc. are used in the control and management of aflatoxin. And several strains of Bacillus subtilis and Pseudomonas solanacearum are used to eliminate the aflatoxin, and also good agricultural practices also help to control the toxins to a larger extent. So coming to the control measures or the management of the aflatoxin production. First one is we can use biocontrol agents in order to destroy these Aspergillus species producing the aflatoxin, and thereby reduce the production of aflatoxin or prevent the production of aflatoxin in a food diet. And the commonly used biocontrol agents are the Bacillus subtilis, Lactobacillus species, Pseudomonas, Ralstonia, Burkholderia, etc. They are mainly used in the control and management of aflatoxin. We know that microorganisms or the products produced by some group of microorganisms can be used to prevent the growth of some other group of microorganisms, and we call it as the biocontrol agents. So the use of the microorganism itself to control pathogenic microorganism is one of the promising method, or it helps to reduce the use of other chemical agents to prevent the growth of such microorganisms. And in the case of aflatoxins, some biocontrol agents, some bacteria species can be used to prevent the growth of such pathogenic fungus and thereby reduce the production of this aflatoxin. And several strains of the Bacillus subtilis and the Pseudomonas solanacearum are used to eliminate the aflatoxin, help to completely eliminate the aflatoxin. Besides this, good agricultural practices also help to control the production of the toxin to a greater extent. Thank you.